So we've seen a little bit about how genes can function, how genes and phenotypes are related to genetics and DNA. And I thought early on in the course we probably should talk about this topic of sort of how do people get in and find out what a gene does or locate a new gene that might have an important function. So we know that organisms are full of DNA and that DNA has functions, but the connection between a phenotype or any other aspect of the organism and its genetics is often something that's is just hidden from us, and we have to have the right tools for discovering it. So we'll talk about the sort of classical version for identifying genes, and also a little perspective on new approaches that are currently used today to find genes. So today's background is this image of some ladybird beetles, and there's a little bit of variation in there. We've got some beetles with spots and some without spots. And so this could be the kind of question you might apply a genetic investigation to see if you can find something causing that difference in your wild population. So starting in with classical gene discovery, I think we often, in these courses, we often sort of are presented with examples that are already well understood. So it's good to keep in mind that there was a time, and there's still plenty of examples of organisms that have interesting features that we don't know why they're there. And this is something that genetics, by and large, can be used to answer. So genetic tools can get in, and if you see something that is an interesting feature or difference in nature, you can use your genetic tools and try to find a cause for that. So um, I don't know, it seems like a whole bunch of the questions about what caused that can be answered by finding the right genetic answer. This is an example. There was actually an article not too long ago about a group that investigated this difference. This is a plant called the California poppy. They actually plant these at UW-Whitewater on occasion, and they've got this lovely bright orange color. And a group of people found, or I guess it's been known for a while, but they decided to investigate, well, what is it about that color? What causes it? So it's known that in some natural populations, you can get some variation like what's shown in this image here, where you have not only the kind of standard bright orange color for petals, but also this pale white or sometimes pale orange petal color. So what causes that? I mean, that's really the investigation starts, and just a good example of science as well, right? So you discover something and you want to know what causes it. You put on your geneticist hat and you get in there and try to investigate. So some questions you might try to get at early on. Again, this is sort of from a classical perspective. So a lot of times in what we call classical genetics, you would take a long time to get familiar with your organism and uh, try to cultivate it in a controlled environment, and then try to do some breeding experiments. So a couple of questions that you'd probably want to address early on in your study to answer these questions of whether individuals are true breeding and whether the trait you're looking at is heritable. So let's see if we can define those for ourselves here. So heritable is a concept that will keep coming up in genetics. And it's just a fancy way of talking about something that you can inherit. So an organism, as we've seen before, has two parents, and this organism inherits something from its parents. You might inherit something from your parents besides your genetic features, but basically what we get for most of our organisms' makeup, certainly when we're getting started in life, we get that from our parents. So you have arms and legs and eyes, and also some individual features like eye color and skin color. These things are determined by what your parents give you. So you inherit maybe not only some I don't know, social status from your parents, you might also inherit, or you definitely inherit your features as well. So if something is heritable, let's say that dad's got um, black hair and mom's got red hair, then you might end up with black hair. And of course, this is something that probably wouldn't surprise you, but to know that a person has a feature that is like what one of their parents has, that is an indication that that is a feature that is inherited from the parent. So this parent has black hair and passed that on to the child. So I guess maybe something that's not heritable, we could use that. Let's say we have an example of, all right, let's give this a try. Say we have something like obesity and not obese. Let's say a person from their parents, they're born to parents who are classified as obese, and then at some point in their life, they reach the same age that their parents were when they were born, and they've gone through their life and they have not become obese. This is something that is not necessarily genetically programmed, and you could probably think of some reasons why that wouldn't be genetically programmed. There's a lot of influence on environment that has to do with somebody's body mass as they grow up. Not to say that this is not completely removed from genetics. There can be genetic elements that control this. And that's also something you get at with genetics, right? Trying to determine which aspects are strictly heritable, being passed on from parents to offspring, and which ones are caused by something besides genetics and possibly through choices, experiences, environment, things like that. All right, so you can determine that these are heritable. One way to look at heritability is to say we've got two white petaled flowers and they pass on their white flowered trait to offspring. So if you manage to cross two white flowered plants and their offspring have white flowers, and then you cross two bright orange flowered plants and their offspring have bright orange flowers, that would give you a pretty good indication that they are passing on something genetic, especially if you grow these in a controlled environment. So you're growing everything in the same greenhouse with the same soil. You eliminate a whole bunch of environmental possibilities for variation. And what you're left with is probably a genetic cause. This other statement of true breeding is also shown on this. True breeding is, for a lot of our purposes in this class, is basically the same thing as saying individuals are homozygous. So I don't know that we've covered that so far, but we'll definitely get around to using this term. What it means basically is if the population of bright orange flowered plants, if they have only the genetic information for making bright orange flowers, then if you keep breeding bright orange flowers by bright orange flowers, they'll keep making bright orange flowered offspring. Same thing over here. If you have individuals that are always white petaled flowers, if you keep breeding them together, that's the true in true breeding, the breed true. Meaning basically that they keep producing this desirable phenotype generation after generation because genetically that's all they've got, right? So they have white genetics for making white petals and they pass that on every time to their offspring. So you've seen this before, I'm sure, but these are some features you can discover if you've just got a new phenotype and you want to know what causes it. You might get in there and try to find out what the genetic cause is, and this is an early step to know that you have something worth studying with genetic tools based on the evidence you have here of them being heritable and true breeding. So let's say we did a controlled genetic cross. This is another tool in your toolkit for classical genetics. You can grow the bright orange petaled flowers up, you can grow the white petaled flowers up, and then you maybe want to discover your next step, find out what happens if you cross a bright orange petaled flower by a white petaled flower. And so what's shown here is one of these controlled crosses along with some terminology that we use to describe them. The first generation crossing these two genetically distinct kinds of plants, that's the P or parental generation. And their offspring is the F1, the first offspring generation. And the F there is filial. You don't really have to know that, but that's the origin. Comes from Latin basically for son or daughter. So the F1 generation is the first generation after crossing these together. And let's say you do this, you cross a white petaled flower by a bright orange petal flower, and you get a pale orange petaled flower. So you got a new piece of information that something is getting mixed, it appears, in the genetics of those two parents combining together to produce a phenotype, an appearance in their offspring that's not exactly like either parent had. And then further on, you could take that additional steps further. Let's say you've got two of these pale orange flowered plants, you cross them together, and one of our interesting genetic results that's held up since the time of Gregor Mendel that we'll talk about is something that these genetic features can show up again in later generations. So if you cross two of these F1 individuals together, some of their offspring look like them in having the pale orange color, and then some of their offspring actually end up looking like the phenotypes of the parent generation. So hopefully a lot of that is familiar to you, and using genetic tools, you could get in kind of under the hood a little bit more and investigate more about these. And this is one that it's a little bit harder to sort of demonstrate this for you. The classical way of discovering something like this involves some biochemistry, some laboratory techniques that we don't replicate in our lab. But ultimately, you basically zero in on something that is present in the bright orange flowered plants, and that same thing is absent in the white flowered plants. So some kind of difference, not just in, well, obviously in genetics, so something different in the DNA of these individuals, but also quite often in the classical sense, you would be looking for an enzyme difference. So something that one of these can do that the other cannot do. It turns out this is the reason. So it is a gene called PSY1 that produces an enzyme called phytoene synthase. And phytoene synthase has the enzyme name there, and it's basically, the name tells you that it's involved in making this molecule called phytoene. And you don't need to know all this, but through this biochemical pathway here, phytoene is a precursor to making some other molecules you might have heard of. Lycopene, that's an ingredient and an antioxidant you find in tomatoes, lycopene. Beta carotene is a vitamin you often find in carrots and other vegetables. And then this other one, zeaxanthin. So basically being able to make this color at the end, these pigmented molecules, is the result of having the enzymes for converting one of these into the other. And the individuals that are missing a functional copy of phytoene synthase can't make phytoene, don't have it as a starting point to make lycopene, and therefore can't make anything else in this pathway. And these are colorful molecules. So missing that enzyme means that the white petaled flowers can't make any of the orangish type pigments that go into the flowers that gives them this white petal color. So we have an enzyme reason. Enzymes related to proteins. Enzymes are proteins, as we've seen in the translation lecture. And so not having the instructions or the ability to make phytoene synthase gives that difference in color. So I wanted to cover this kind of early in the course because this is really our current fundamental understanding of much of genetics, is not just some sort of magical reason, gene, DNA present, color present, but it's actually a DNA causes enzyme, and a different version of the DNA causes a different version of the enzyme, or a missing or non-functional version of this enzyme. Okay, so the non-functional version of that enzyme comes from having a nucleotide sequence to make that enzyme. So the DNA that encodes the protein is different between these two kinds of flowers. And it turns out that one of these, the one that ends up with the white petals, has a stop code on here and makes this terminate, this whole protein terminates early and actually in the middle of the active site. So it's not able to do its job as an enzyme because it doesn't have the correct programming to make the amino acids that are part of that active site. So you kind of put all these pieces together. We've got inheritance evidence, you've got biochemical evidence of what enzymes are present, what pigments are being made, and then here we've got DNA evidence of what the sequence is for encoding that protein. All right, so I've highlighted these here just as different alleles. So PSY1 plus is the functional wild type version of the PSY1 gene, and PSY1 minus is the non-functional or missing or broken mutated version of the PSY1 gene. And then of course, because these are plants, I don't know if you knew, but plants are diploid. So flowering plants are diploid, and they get 2 copies of the PSY1 gene. So these populations that were true breeding that we saw before with the bright orange being able to continually produce bright orange offspring, that was a population that had only the PSY1 plus. So diploid individuals would be called homozygous, PSY1 plus, PSY1 plus, homozygous, well, I guess we'll just stick with that. Homozygous for the plus allele. And the population that had white flowers is homozygous for the minus allele. So no ability to make the orange pigment in the minus allele homozygous individuals, and full ability to make the orange pigment in the homozygous plus allele. And knowing that, we can understand what happens in the F1 generation, where a combination of the plus from the bright orange parent and the minus from the white flowered parent combine together. And this is one possible outcome, is that this phenotype of the F1 individual doesn't look exactly like either parent, and biochemically it's making about half as much or a reduced amount of the pigment, and so a reduced amount of pigment gives you more of a pale color. So it's got the ability to make this phytoene synthase enzyme, but it doesn't produce as much because the copy number of the DNA is related to the abundance of the enzyme. All right, so coming back around, we're going to get to the predictions of Mendelian genetics in a later lecture, but you can lay it all out here just without thinking about the ratios and Punnett squares just yet. You can put on there the genotypes of the F1 individuals. These are heterozygous plus and minus individuals, and it's possible based on how they combine from the male parent and the female parent that you could get a minus from one and a minus from the other. That's possible here. A plus from one, a plus from the other, that's possible here. And a minus from one and a plus from the other, and that's possible here. So it's possible to get different colors in the offspring, and if you understand how that all gets put together, you can predict this, and you can understand how these different colors would show up. Okay, so just an overview of what I'm calling here classical gene discovery. Starts off with identifying variation in nature, and then the second point here is maybe to induce mutations. So a lot of times if you're trying to study, a lot of times people would study as much as they possibly could about an organism. And one way to gather up more variation is to actually mutate, deliberately alter the genetic content of individuals. So you could deliberately mutate them and see what kind of phenotypes you generate, and then go through this process of figuring out why the particular mutants cause the certain phenotypes. And then as the second big bullet point here says, as I've said, conduct genetic crosses, biochemical investigations, etc. Find the molecular causes of the phenotypes. And once you can basically say what the gene is, as in where is it in the genome, and what it does, how you translate DNA into phenotype, then you've pretty well genetically understood the basis for the phenotype. And just another point about this is that being able to study model organisms, such as the fruit fly that we're doing in lab, it gives you a lot bigger toolkit for understanding the genetics of organisms. So a few advantages is that you're maybe comfortable working with an organism. You've got a reference set of information. So this is just an illustration highlighting a paper that um, shows the structure of chromosomes of these fruit flies. And you could put a newly understood genetic component into the context of this map of, say, you got a map of the chromosome where all these genes that have been discovered show up on that physical piece of DNA, that big long chromosome, figuring out where they go, and then also this information about what they do. So lots of advantages to working on model organisms. And I would say genetic investigation was sort of indispensable early on to be able to study a well-known organism and put your new discoveries into context.